Rose. I'm, for those who haven't met me before, or I think I've spoken to nearly all of you on email. So I'm a Local Nature Partnership coordinator. So I look after the mailing list of the Local Nature Partnership and send out various events and details and coordinate the secretary and secretariat and so on for RCT LNP. So RCT Local Nature Partnership is a partnership made up of various recorders, groups, organisations, all taking action for nature in RCT. So there's quite a few here today. There's Glamorgan Fungus Group, Glamorgan Bird Club, BTO Regional Group, Emma's here from Colespoil Fungi, Marie from the council, and all these other organisations or groups have got you know, a part of the partnership. They're all doing stuff for nature in RCT. And just to quickly go through what we do, yeah, basically we work together to plan and deliver action for nature in the county. And we try and raise awareness of the nature of RCT, we record the nature of RCT, protect the nature of RCT, that's what we're all here for. And we try and manage areas as well for the benefit of nature. We also do some plans and policy development, networking. That's a good place to network. We meet every 3 months, or sometimes a little bit more frequently as well. And you can meet up with all these other groups and organisations. Practical conservation projects and reporting, education, awareness raising and advice as well. So that's what the partnership is here for, basically. And at this point, I just wondered if anyone wanted to give any update on their group or what they've been doing in their areas. Yeah, Pontclean Environment Group, I know you've been up to quite a lot because we've been doing stuff together.
Yeah. So I've popped in the chat as well that we've run 2 events over the summer with the help of the Local Nature Partnership. Back in July, we had a guided nature walk leading from alongside the river up to a pant marsh. And as a community group, we're all enthusiastic. We don't have the expertise. So it was great to have Rose and Richard along for that one to talk us through some of the key points and of plants, wildlife, general habitats. It was brilliant. And then just last week, we were led again by Rose and Richard for a bat detecting evening, and we had really good levels of interest for that from the local community, and we had a waiting list as well. So it's been a great way to engage more people in nature and do something for all ages. So thank you.
Anyone else been up to anything they wanted to share or wanted to stick in the chat if you haven't already? Alan?
Hi, is it worth mentioning the SWIFT project that we're looking at in Ferndale at the moment?
Yeah, definitely, yeah.
I've had a few your emails with the contact from the school in Ferndale. Do you want me to go and contact Richard directly?
Up to you entirely. If you wanted me to sort of be the go-between, I'm happy to do that.
I don't mind, because it will need a site visit.
Yeah, I think so.
For the others that may be listening, we are hoping to put some SWIFT boxes up in a school in Ferndale, and it's just looking at the possibilities at the moment.
Yeah, we did a site visit earlier in, well, when was that? July time, was it? Yeah. Looking for swifts, and it seems as if the population has declined dramatically up there. So I'm hoping to do some work for them. Yeah, see, Marie Claire just put in the chat about natural nesting sites, or maybe unnatural nesting sites in houses are being lost at a rate of knots as people do up their fascia boards and things. So yes, we have to see if we can help them somehow. Thanks, Alan. Anything else on that?
Not for the moment, no.
Great. Brill. Okay. Anything else? Put it in the chat as we go along. That's fine. Brill. And then this is something that the partnership as a whole have been working on recently. which is our Action for Nature plan. So this is something I had an email from Helen actually recently asking for objectives that us as the Nature Partnership think might be the most beneficial for community groups or other people active in their communities to do to help nature. And this is what we're working on at the moment. Basically, this is our Action for Nature plan, which is under construction and being written at the moment. There's lots left to do on it, and I've put the web address there, which would be really good for you to go and have a look at. I will actually share my screen if I dare change screens and show you briefly the idea of it. That we've got a different part of it this time. So this has been is under review. Basically, it's been written before, and it's now being updated. And there is a section, if I can share my screen again. Right, so this is the site when you come onto it, and there is a section here which is called How Can I Help? And what we've done is sort of put different groups of people in here. Again, this is all under review, and you might think that there's a better way of grouping this, and actually we'd really love your feedback on it. But for example, if you were a family with children looking for something to do, our idea is that you'd be able to go to this page, and it'll give you some ideas of things that you could do to help wildlife in your area, learn more about wildlife. So it'll start with these actions. This is a video about dormouse nut hunting. We'll talk about that later. And that's the idea of these pages, basically, to give you a really brief idea of some things that you could do. So for example, businesses, you'll see it's not written, we haven't finished it, but to give you some ideas of how you can help wildlife of RCT in particular. So I just wanted to show you that quickly, just to get your feedback really, and see if you thought that's a good setup. If if you wanted to go off and have a look at it, as usual, I'll send round the slides afterwards. Can you see the screen again now? You should be brilliant. Thanks, Marie. Yeah, if you wanted to go and have a look at that website, we'd be really grateful if you wanted to have a look and see what you think of the layout, basically, and if it's something that you would find useful, or if you've got any ideas of how we could make it more useful. That would be brilliant. So I just wanted to mention it at this point, and show you how it works. So we are. Wayne, did you want to say something?
Yes, it looks great. Very quickly seeing it there, Rose. A couple of points, I guess. One would be how groups could get their own content on there. And two, do you have a calendar of events or plan to?
That's a really good idea, actually, for groups to be able to add their own. So we'll have to think about that. I think we will have there's a tab here for the Nature Partnership, which will have a list of the groups that are active in the partnership as well. And maybe, we could have some sort of way of you updating things on the website for yourself. That would be quite good. Probably we will have a news section as well for news articles, and then calendar of events. We'll have a think about that one. It would be brilliant. We'll have to see how it would work and if we are able to keep it updated. So, yeah.
Sorry, Rose. This is where the issue might be with us is actually keeping that calendar of events updated regularly and maintained and stuff, because it's hard enough for us to do it on our current websites now, and try to manage multiple sites might be difficult for us. It's a good idea, and I would love to have it, but we would have to think logistically how we would manage that space and afterwards.
It would be a good thing to have. I wonder if we could link it to a current sort of events calendar, have it embedded on the site so that people can add their own events. Maybe that would work. Anyway, we'll think about that one. Thanks, Wayne.
Shubrek already do it actually, but perhaps the link into Shubrek. I don't know. I haven't looked at theirs recently.
That could work. Well, they're supposed to be having their website updated as well, aren't they?
It's the whole of the South East Wales area, but this would be more RCT, obviously.
Yeah, no, that's a good idea. Mark, you've got your hand up.
Yeah, Rose. Some of the text, the colour and the font and font size is a bit difficult for me to read.
Okay.
And I'm sure a lot of other people would have problems with it. So I think perhaps you ought to look at.
Yeah, accessibility.
Yeah.
All right, we can do that.
The colour's a bit of a problem, and the font perhaps needs to be increased. But
that has been mentioned before, actually, the colour. So we'll work on that. Thanks, Mark.
Okay, cheers.
Brill. Yeah, any other further feedback that if you've had a chance to go away and look at it closer, just email it to me through my email's at the end of this presentation, but we'd be really grateful for anyone to take a minute to have a look. So moving on then, this is something that we usually try and have a little section on awareness raising in your community. So for now, this is the perfect time to mention this about essentially nature isn't neat, isn't it? What Monmouthshire say, or Newport Council, I think. So at this time of year, obviously everyone's very tempted to go out and tidy up their gardens, tidy up green spaces. And what perhaps they don't realise is that, patches of plants like this, where you've got lots of dead tall stems, are really good hibernation sites for numerous pollinators, bees and butterflies, etc. A lot of them will overwinter in dead plant material like this. So actually, if people are able to leave a patch of this dead vegetation standing, and then it would be really helpful for the pollinators, and you can then clear it away in the spring once the temperatures are consistently reaching above 10 degrees C, and that just enables the insects that are hibernating there to sort of wake up and fly away before you clear things up. So that's a really nice thing to remind people at this time of year before they start getting trigger happy with the green bags. And then obviously ivy as well. I'm sure many of you are aware of ivy is a really important it's autumn to early winter pollen source and food source for these late invertebrates. So this bee in this picture is quite a newcomer I think it's new to the British Isles, or it's moving more further north. Someone might correct me on that. But that's the ivy bee, and that's one that you'll see at this time of year, well, later in the year now, nectaring on ivy when they come into flower. So that's a really important one to leave up. Make sure that people don't clear away their ivy too soon. So those are nice things to raise awareness of in your communities, if you can. So moving on then to some ideas for events and activities for the autumn and the early winter. So you can suggest them for people to do. You can do them yourself as your group. People can do them in the garden, or they can go out and about, or with a group, or however. So I've done my best. It's a difficult time of year, October, November, December. Things do slow down, and there's less surveys and things to do, but there is still things you can do. I've started with one here, which perhaps is getting a little late for it, but you could definitely start this in the coming month in September, and perhaps if the weather conditions are right, you might have luck in October as well. And that is to record Orthoptera, so your grasshoppers and your crickets. So hopefully this video will work. It's just a little video about an application that you can use, a phone app. But if you go to sort of patches of grass that haven't been mown yet, I mean, we saw at Helen the other day on the bat walk, the bat detectors were absolutely heaving with the sound of crickets, weren't they? We possibly heard more crickets than bats. but this is a really nice one to use, and this app is really great. It's got identification tips on it, and I'll just see if this video works. And it might. Oh, there you are. It's going to work.
The grasshopper record.
Can you hear that? The volume.
Yes.
Brilliant. Okay.
The scheme has been running since 1967, and it's
We can't hear it now.
I muted myself. Okay, you're hearing it from my screen. There we are.
been seen over the years, and now we can look at changes in those distributions over time, and the app will help people to make observations more easily, and we hope send more of them in years to come. There are about 25 species of grasshoppers and crickets in Britain, and the related species. So there are 4 species of native earwigs, and 3 of native cockroaches, and then 2 naturalized stick insects. Crickets and grasshoppers are easily told apart by the length of their antennae. So grasshopper has short antennae, and this is a cricket where the antennae are basically as long as the body or longer. The best time to see crickets and grasshoppers is late summer, so generally we sort of say when the birds stop singing, the grasshoppers start, so late summer, July, August, September. And good places are any grasslands, so meadows with a little bit of tussocks or wildflowers that aren't cut too frequently, or roadsides.
Yeah, so if you follow that link when I send the transcript around, or if you just search it in app stores, what's it called? I Record Grasshoppers, and it's a really good app. But yes, one for sooner rather than later. I'm pushing it a bit, putting it into this to this one, but I just thought it was a nice one to get out and do now. Quite an interesting one that people wouldn't necessarily think about. So getting into something that is definitely around in October, we've got Saturday the 2nd of October this year is UK Fungus Day. There is a Welsh Fungus Day as well. I don't know, Mark or Emma, if you know the date of that one this year. Do you know?
Yeah, is that at the National Botanic Gardens, Wales?
Mm.
Yeah, no, I haven't seen anything about that one.
No.
I'll do a plug here for the Glamorgan Fungus Group and the Kenbig National Nature Reserve. Chris Jones, who's the warden there now, he's organising an event for the Saturday the 2nd of October, and Glamorgan Fungus Group are going to be, well, if all goes to plan, we'll be attending there and setting up information, photos of fungi, and trying to encourage the public to be to understand a bit more about fungi, the forgotten kingdom, and hopefully we'll get quite a lot of people coming along to that. It's all a bit dependent on COVID regulations and how they move. I think there will be, Chris will be leading some forays out on the nature reserve, and hopefully we'll be in the visitor centre with a lot of information and be able to talk to the general public and try and raise the profile of fungi.
Brilliant. Sounds good. Yeah, so it's a good day to get involved in. There's just so much interesting things about fungi, isn't there, that we just don't have a clue about. So on that note, Emma, did you want to jump in?
Okay. Did you want to do the screen share, Rose?
So, do you
Or
want me to do it, and up to you completely.
yeah.
Go on then.
If you wouldn't mind, that'd be great. Thank you.
No worries. I'll change my PowerPoint. There we go. I need to move it. Over here, would you? Oh, bear with. Right, I'll have to share it on this screen, and I won't be able to. I might not be able to see you, but I'll go for it. There we are. Okay.
Okay. Right. What we have here is just a very quick introduction to fungi recording. Not going to cover the usual elements of different fungal groups, such as Basidiomyces, which are the spore droppers, or the Ascomycetes, the spore shooters. This is more to do with the what people need to be looking for that brings them a step forward to be able to identify what they actually have. Because it's very, very overwhelming. From my own experiences, when you're actually teaching people field work, is that it's just too much to take in with fungi, because it's a massive, massive diversity. It's its own fungal kingdom, you know, so it's huge. It's absolutely huge. So this is just the pointers of features that will actually help you get to ID. They're absolutely vital to helping you learn as well. So we'll go on to the next page, Rose. Okay, and we're not going to discuss either anything other than this is for the purpose of biodiversity recording for the preservation and conservation of fungi and their habitats as well, which in RCT, as in the whole of the UK, fungi are not very well represented and are not a priority in many cases in any land management schemes. So by getting people on board to actually find fungi, record fungi, it actually gives decision makers a better understanding of what's in the area as well. Next page, please, Rose. Okay, so the fungal kingdom, it's absolutely immense. In the UK alone, we have over 17,000 species, and that list grows weekly, monthly, yearly by quite a large number each year. Estimated that there are 3 to 5 million species globally, and they're the largest kingdom. The earth, it's absolutely vital for the survival of the earth. 95% of plant life are reliant upon fungi in a mycorrhizal relationship, which means they're symbiotes. They work together, bringing those plant life nutrients and water in exchange of sugars. And they also have another element where they can actually prevent plants and trees from being grazed by animals as well, like an insecticide by taste in particular. It deters grazing as well of certain plants. The fungal kingdom has the largest living organism at the Amaryllia ostoi, which is one of the honey fungus in America. It's actually, the size of it is 13,000 football fields in size. That's absolutely huge, and it's some 3,000 to 5,000 years old as well. So fungi can be quite large in their mycelium mat underground, not the actual fruiting bodies, the mycelium mat that we don't see, but it's underground. They clean up and break down pollutants in an environment. If they currently being used to clear up oil spillage, particularly in Cuba and other areas where they have massive pollution with oil, the fungi are being used. Oyster mushrooms is one of the ones that are actually used as a cleanup, and they've also discovered fungi that can break down plastics, not in months, but actually break down plastics completely. They consume it and break it down within a number of weeks. And they are nature's natural cleanup team. They actually break down all organic matter, be that plant life or even bodily, animals and even humans. They can actually break down everything from bone, tissue, keratin in hair and nails. That there is a species of fungi that can actually do that job. They're also the only organism that can actually break down lignin, which is the cellulose type material that's actually in trees. And without fungi, the world would look very, very different, as we would not, the trees would actually fall and not decay. And that is the history of coal. It's actually produced from when fungi actually learned how to consume trees. The actual process of coal then ended in the timescale of things. That's another story in regards to coal. But the list goes on and on for the wonderful things actually fungi do for us. If we go on to the next page, please, Rose. Now, when we're actually looking for fungi, one of the first things that we must do is actually take note of the habitat they actually reside in. It's really important, as you have various groups of fungi which will actually reside in each of their own habitats, and taking a note of that will actually help you a long way in your ID. So make a note of its grassland, be that meadow or open grassland. Woodland, is it coniferous or is it deciduous? Because you have certain mycorrhiza fungi that will only actually work with either of those two. Is it peat bog or wetland? Fungi will actually survive in very arid, dry conditions, and also very wet. They don't mind getting their feet wet if they're a particular species. And you even have aquatic fungi that actually live in water. Is it dune or coastland? Upland or high altitude? Some of the rarer species of fungi are actually found in a high altitude. In Scotland, they're currently doing some amazing research at the moment using soil samples. So that's going to produce a huge number of new to UK species. Scrubland, absolutely amazing for fungi, very much overlooked, especially in land management. People sort of look at a big bramble patch and go, oh, it's bramble. It could actually produce an amazing array of fungal species. Urban settings, industrial and brownfield, these are the ones that throw a spanner in the works, because they have mosaic habitats and very fragmented habitats where conditions can have a massive variant within meters. The habitat can change drastically. So it's really important to be aware, are you on a brownfield site, a post-industrial site that's been either just left to actually regenerate on its own, or if it's had a lot of work done on it. And when they have work done on them, those trees are brought in and soils are brought in, can bring in lots of fungal species as well. So very important to mention if you're in an industrial or brownfield site, because your species will turn up very, very unexpectedly that may seem as if they don't belong there. The next page, please, Rose. What is your fungus growing on, or what is it growing with? Very, very important. You need to make note of substrate. Is it dead wood, moss, bare ground, fallen leaves, cones or debris? What tree species, if known? And also on the tree, what part of the tree is it growing on? Is it growing on bark? Is it growing on bare wood where the bark has been stripped away? Is it growing on a cut end or a cavity? All those little extra information are vital. What plant and what part of the plant? Is it on the upper or the lower side of the leaf? In regards to moulds and mildews, they can be quite specific. Some grow on upper, others on lower, and they can actually split your species up for you. And also it's growing on the stamen, which were then in the realms then very often of your smut fungi. If on the ground, what trees or plants are the nearest? Because you have your mycorrhizal fungi, finding out the nearest trees again will actually help you get to an ID. And keep an eye open as well for seedlings, because you may think, oh, there's only pine trees around there, definitely wasn't any birch. But when you go back and check, you'll actually see some birch seedlings, and you only need the presence of a tree. Sometimes they can be dead trees, other times not so much. If one of the species that we're going to be losing in RCT is species such as the larch bolete, because we're having our larch trees felled. And those species will only live for 2 to 3 years after the trees have been felled. So we probably will see quite a drastic change in species in RCT over the coming years. And next page, please, Rose. The next thing we need to look at is what type of fruiting body. Does it have gills? Does it have pores? Next page, Rose. Is it toothed? does it have pseudogills? Now, pseudogills, unlike true gills, are more like vein structure, hence why they're called pseudogills. And a very, very important species, such as the chanterelle family, the Cantharellus, which have the pseudogills. Next page, please, Rose. Also, is it resputinate or is it crust? These are species that tend to grow horizontally across wood, but they can actually have, very confusingly, a shelf or bracket type formation, which can actually confuse you a little. But again, you're looking at the larger structure, so it should be growing pretty much like a crust spreading across the actual wood itself. Then we have the bracket fungi then, or your shelf fungi. Next page, please, Rose. Your coral fungi. Now, these ones are a little bit more confusing because they have a combination of species within this group. You have Basidiomyces, which are your spore droppers, and your Ascomycete, your spore shooters. You can't always tell by looking at these which one you have unless you're using a microscope, unfortunately. But again, by taking note of habitat, the structure, the way the arms are actually branching off, how many fingers they have, colour and smell can actually help you get to your correct, at least correct genus. And then you have your earth tongues, which vary in colour quite drastically, from blacks all the way to olives and greens and purples. And these ones are microscope, but it's better to have a record of earth tongue rather than no record at all, because that can alert people that that habitat is very promising, as you will only get earth tongue in generally land that it's very quite mature. Around the 30-year mark is when grassland, especially when it's very moss heavy or bryophyte heavy, they start having a greater species list from around the 30-year mark. So by spotting earth tongues, that is a very good indicator of older soil, more mature soil, and also likelihood presence of wax caps as well, which then raises potentially the status of that habitat to be in an area of interest that needs more survey work. Next slide, please. We then have your jelly fungus. Now, the jellies are consistently like a jelly. They can grow, this species here, salmon fungus, grows on the ground in association with pine trees and some other coniferous trees as well. But majority of the jelly fungi are actually present on trees, such as the amber jelly, which grows on birch or willow, and quite a striking species. You then have your cup fungi, and these are Ascomycetes, and these particular ones, again, most of them are microscope job, but they, at least this one being the scarlet elf cup, it's look like being the ruby elf cup, but they are, it doesn't matter which one, if you actually record the scarlet elf cup or elf cup, that is fine. And again, it indicates again that it's an area of more maturity. What I have found from my own observations, and mine are checked by microscope, is if you're finding scarlet elf cup only, you're in acidic ground, and if you're finding alkaline ground, then you're then getting the rarer ruby cup, which is a very nice one to find. I've not found ruby as yet within RCT. I found it just on the borders, unfortunately not within RCT. That doesn't mean it's not here. It means it's simply not yet found because we haven't got enough people using microscopes to determine exactly where it is we're looking at. But this is one particular, you'll find the scarlet on coarsefoil, and in our wet woodlands throughout RCT, as we are generally, we are acidic ground. Next page, please, Rose. Then we have our earth balls or our puff balls. Now, these groups do confuse people quite a lot, because when they're immature, the earth ball and many species of the puffball family look very much alike. In their mature form, they actually have the same purpose. Their inner gleba, their innards, will actually become a powder, and then they'll rupture either by wind, rain or touch, and they'll actually disperse their spores. But when they're immature, you can actually split the earth ball and the puffball family by simply cutting through. And on the earth ball family, all of them will have this dark gleba internally, whereas a puffball will have a white internal body before it starts maturing. And they also have a distinctly different smell. The earth ball family have a chemical, I think it's pronounced udin, which is a chemical they also use in the perfuming industry. It's quite pungent, quite strong. And once you get your nose around these things, you can actually detect an immature earth ball, which hasn't yet gone dark in the centre, from also puffballs as well. And again, with the earth ball family and the puffball family, they can be quite difficult to get to genus. So if you're starting out on recording and fungi identification, it's a massive part on the back just to actually differentiate. You've got an earth ball, a scleroderma SV, or a puffball, and then, you know, at least you're on the right track, and you get help then further on to ID to actual to get you to species level. And the next page, please, Rose. And again, still within the sac family, we have the earth stars, which are a funky group of fungi. They sit on, they start out like an onion shape, which then opens up and splits to actually form these arm-like projections, and the sac and the spore-bearing fruiting body sits on top, and again releases its spores. And we have the most common one we have in the Rhondda is the collared earth star, but we do have some rarities as well, which including the beaked earth star, which is one in the image there. And you'll find this one in some of the cemeteries in RCT, and also the only colliery site I found the beaked earth star so far is at Tower Colliery. Can't find it anywhere else. And just added on there, it's not really part of the sac family set, but it's the bird nest family, because these are very, very little fungi. They have, this is the fluted earth, the fluted bird's nest fungi here. And within the fruiting body, you'll notice little white eggs, and they actually, when the rain hits them, they actually bounce out, and that's how they actually spread their spores. Quite a large genus there as well. Most of them can be ID'd fairly easily, providing you can actually see the actual internal egg structure and the colour of them and the number of them. And with the fluted one in particular, this one, even when the eggs have actually been dispersed, it's so distinct in its flute-like, almost trumpet formation with very heavily striated internal. It's a very easy one for beginners to actually identify. And you'll find it quite a few places in RCT. It generally prefers older woodland, not necessarily ancient, but certainly veteran woodland. And you'll find it pretty much only on deciduous woodland. I have found it once on conifer, but that was very decayed conifer. So I think that was, you know, an oddity rather than a regular thing. Next one, please, Rose. One of the most important things that you can actually do to actually help you along with your ID and to get further help is to ensure that you photograph all visible features. With the gilled and pored fungi, the typical mushroom or toadstool shaped fungi, you're looking to get good clear images of cap, stem and gills or pores, including the base of the fungi, because the base can tell you very important information, be it bulbous, rooting, if it's actually got mycelial cords coming off it, or sometimes even a changing colour. So they're very, very important features. I'm not the best photographer in the world, and in fact it's something that we always talk about, but you know, generally, even if you're not good at making photographs, as long as they're not too blurred, then they will actually help you no end in getting further help towards your ID. And also they're vitally important when you're entering records. We need images to actually be able to verify what it is you're actually having. If you've made a mistake, we can actually help guide people further into getting the correct ID. And the next one, please, Rose. And then another really important feature, it doesn't change colour when cut or bruised. Now, the one on the left there, that's Cortesium roseum, which UK listed is very rare. Now, we're quite privileged in the Rhondda because of the whole of RCT, because of the influence for coal spoil and the microhabitat which you tend to have in these habitats. is that this species is exceptionally common. You won't find a single coal spoil in RCT that doesn't have it, and neither will you'll be hard pressed to find a stretch of river line that has willow that does not have this species. It's a stunning pinky colour, anything from very, very pale just pink right the way through to blushing pink. And you easily determine the ID on this one simply using pressure. And on this image here, you can see quite clearly that port red stain in the centre of the image. That was simply done by applying pressure from my thumb. So it changes colour quite rapidly. And it's a very, even though it's common in the RCT, every record of fungi is important, doesn't matter how common it is. Again, because it's a rare species, it also encourages others then to go out and find it as well. One of the other ones with bruising are the bolete family. Now this one is Sulius gravelli, and it actually stains a very dark brown, orangey brown colour. And just for a bit of fun, when I was actually doing this one, I just wrote coal spoil in there, as this one was actually on the bulk coal spoil site. So just a bit of fun there. But a very important feature, what colour they actually bruise. Some will actually bruise blue, greens as well. So the colour is very, very important to actually make note of that. Not just in the cap itself, but also in the stem as well, which can have different bruising from the actual cap. And the next one, please, Rose. Now, this one actually surprises people. And providing you have a decent sense of smell, you'll be finding actually learning these. You can split fungal groups on smell alone once you get the hang of it. But you can also get some species to ID simply on the smell alone. And when you ask people generally, what does a mushroom smell like? You tend to get the same reply. Oh, is it earthy? Is it mushroomy? And that's it. And when they actually discover that the list of smells is actually huge, they're blown away, especially when you're actually able to hand them a mushroom that smells of apples, such as there's a Russula quitelli that smells of apple. Sorry, quitelli smells of gooseberries, and there's another Russula that smells of apples, stewed apples. We also have bleach. There's lots of the disc family have a smell of bleach, is quite unpleasant. Marzipan or almonds, cucumber, chocolate, pelargonium, and pelargonium. There's a Cortinarius pelargonia that is split from its lookalike, the frosty webcap, on that smell alone. And to get an identification on a Cortinarius in the field is a magical moment, because they're one of the most difficult genus. And even with the microscope and chemical testing, you'll end up crying because you find out they need DNA sequencing. So if you find a Cortinarius that smells strongly Pelargonian, then it's a bingo, it's a winner, and it's amazing. Get it recorded. You also have flower. Lots of the groups smell of flower. Tricholoma family and the Clitocyber family generally are the two main groups known for a flower aroma. But again, once you get your nose in, you'll actually be able to distinguish the different other undertones and chemicals in those groups as well. Coal tar. Now, coal tar is quite a surprising one, but you actually have one on coal spoil, and lots of waste ground that smells strongly of coal tar. Fish, not the most pleasant at all. Some of the Lactarius family and the Russula family have a very strong fishy aroma, and it is, if you have want to leave one of these in your pots, it's very unpleasant. You also have leather and soap, lemon, citrus, feces and decay. Again, not very pleasant, but that's your stink corn family. But it actually has a very strong floral undertone. So don't be put off by initial smell. Go in for a second and a third and a fourth smell once you start gagging, because the undertones are actually there as well. But the list, and that's just a tiny number of the list of aromas that fungi have. It's very important, wherever fungi you actually find, using nose, have a really good smell. The next one, please, Rose. What colour? Now, this is quite difficult because if you've not produced an actual photograph and it's a photograph that has true colour, having a conversation with someone about what colour your fungus is quite difficult, because one man's mauve is another man's purple. So it's very, very important that when you're taking photographs of fungi, take them in natural light. And if you're aware of any colour that's giving you a flashback, I tend to wear neon colours when I'm out for safety, and the blowback off an orange jacket I was wearing last year was absolutely horrendous because everything had this warm orange glow, and I didn't realise until I was getting home and it was images were ruined from this warm orange glow. So again, is double check, are there any other colours around the mushroom is actually changing the natural colour, because a subtle change in colour can actually throw you off your ID efforts. So good images, and take a note in the field as well in natural light. Next image, please, Rose. Now, once you've actually, if you're feeling more confident within your fungal identification, especially with your guild fungi, you're going to need to look at other features, the stem, the cortina, which is the webbing on families like the Cortinarius, not restricted to Cortinarius, and this will actually throw a lot of people, because the other groups of fungi also have a cortina. The Tricholoma family in particular, a few of them have a very woolly cortina, which will throw you, and Inocybe, which is a relative of the Cortinarius anyway, lots of those have a webbing around the gills. So you have to look at all other features as well. When you're looking at them, do they have a ring? Do they have a rooting element to the stem? Does it have a vulval sac, which is then you're in the Amanita family or the Vulvera family? So again, lots of features that you need to look at on the stem itself, the shape of the stem, what it looks like, and again, take notes, but also take very good images. Lots of these features will actually break away, maybe if you're taking a specimen home, they may break away. They also change with maturity. So a species that may have a large, lovely pendulous, almost like a petticoat going around the stem, can actually break away within the first 24, 48 hours of emerging, and that might be a vital ID feature. So you'll need to ensure that you've got all the other features as well. And next slide, please, Rose. And the gill attachment is absolutely vital, and one of the main ones that people to learn straight away is whether a fungus has a decurrent stem, which means it's running down the stem, or if it's adnate, which is just tucked in off the side there. But all these features are actually, you get the diagrams in any good literature book. Take a book up with you, try to learn them, but better still, bring a specimen home with you. There's plenty there. Make a cut through the centre and you can clearly see the attachment. It's not as difficult as it sounds, especially with all the imaging, but it does take a knack, it takes time and it takes patience. And once you actually understand what each term means, you'll actually be more confident when you're in the field in making that decision in the field as to what type of gill attachment you actually have there. Next one, please, Rose. I'm rushing through because we've only got 20 minutes. One of the other features is, does it bleed or does it produce latex when cut or crushed? Now, one of the main families that produce the milk and the latex are your milk caps, the Lactarius family. And the milk on these, which will be on the gills when they're broken, or the stem when you cut through, and the cap, the colours vary from white all the way to bright oranges, greens, reds. So it's important to make note of what colour the latex actually was, not only when it was first cut, or what colour it actually became later. This particular species here, initially starts out bright orange there, and you can maybe be able to just see on the stem there to the left of the stem that's been cut, it's starting to turn a pinky shade. Within 20 minutes, that whole cut through section, and I couldn't find the image to go with it, the 20-minute later one, but that whole stem there was bright port red. So again, it's important to make observations, not straight away, come back to them 10, 20 minutes later, if you can take a specimen home to do some of these checks with. This one, the shag ink cap on the left-hand side, it's a little bit of a trick because lots of people think that's bleeding. It's not. That's guttation, which is lots of fungi can actually do this, and they actually simply push out excess fluid, and the excess fluids can either be coloured by chemicals within the fungi itself, or if they're grown on a tree, they can be coloured up by the tannins from the tree as well. So this one was a shag ink cap, as I said, and it was actually blood red droplets. One of the most amazing things, and that is a natural phenomenon with shag ink cap. It's a rare phenomenon to see, and generally to see this happen, you'd have to see them just before they actually start to open up, and early in the morning. I went out at sort of 5:30 in the morning to actually catch this group of shag ink caps actually doing that to capture it. The next one, please, Rose. Now, the recommended literature to actually get you up and running with your ID, the one that I recommend to absolutely everyone. I still use this one in the field as well. It's always I've always got a copy in the car. It's a Collins Complete Guide to Mushrooms and Toadstools. Within that booklet are very, very good clear photographs of fungi. The reason I like this book so much is because it gets you in the habit of looking at habitat, tree association, and all features. Also in there, you'll actually have diagrams of gill attachment, and lots of, there's basically anything that you need to know to get you up and running in Fungi ID. It's in this book. It's written in plain English, clear English, and this is as good for young children as it is for the mature adult, and it's very easy to use and follow. Once you've actually moved on a little bit more, and a little bit more confident, then move on to the KB guides. Now, these ones actually use keys. They also include microscopic information, taste testing, and lots of information on, you know, the spores and microscopic features. So it's a little bit more technical, but the keys also include the best macro available. So you can get so far with the ID as well by using the macro features on their own. Next one, please, Rose. Also fascinated by fungi. This book is so much fun. It's, again, it includes everything that you need to know. Includes a lovely section on photographing fungi, which didn't help me because I'm just rubbish at taking photographs of mushrooms, not to how much I try. But it's a really good section on photographing fungi as well. Again, microscope fungi, all the ID features are needed. Excellent section on conservation of fungi and habitats as well. And it's also written in such a fun manner. It's absolutely brilliant. Pat's a really wonderful chap as well, and he's more than happy for people to contact him directly as well for any help that you might need with your Fungi ID. And he has an amazing website, which I forgot to put on here, but Fascinated by Fungi, and I can actually send a link later to Rose, and it's very, very good to when you're stuck in an ID, gives you habitat, features, lookalikes. So if you're a little bit torn and you're not sure if you've got a Russula or a Lactarius, then that website will actually point out the similarities, but the key difference in the two as well. So it's very, very good website. And using, for further ID help as well, using your photographs and all those notes you've taken, habitat, colour, smells, all that information. Then you can upload that information then onto the British Mycological Society page on Facebook. They're very, very helpful. You're hands-on with national experts and worldwide experts as well. You've got the likes of Geoffrey Kibbe on there, and the overall some of the UK's leading mycologists, and they are approachable and they're helpful and they're friendly. So don't be put off thinking, oh, it's, you know, it's only for mycologists. It's not. The British Mycological Society is there to help everyone, and they don't care if you're right at that beginning of your journey and you simply don't know the difference between a pored fungi and a gilled fungi. They're there to help. Locally, you've got Glamorgan Fungus Group. Again, very helpful and informative group. Lots of experts on there. And again, it's the same principle, upload your photographs, good clear photographs with all your information for your help. And if you're stuck and you feel a little bit shy and you don't want to be on social media asking questions, by all means, you can send me an email. I'm on downtime at the moment, so my Facebook pages aren't active, but you can email me if you're absolutely stuck, and I'll help you. And the next page, please, Rose. And finally, why record fungi? Basically, it's essential. We need it to map their presence for better land management practice, and that land management practice includes things which are not seemingly that obvious, such as the way our cemeteries within RCT are managed for waxcap, the Hygrocybe family, how our meadows are managed when they're cut, things like our verges that may be overlooked and have a wonderful collection of fungi growing on them, and our woodlands, because they're not often surveyed, and we're also losing ash trees hand over foot at the moment within RCT and the whole of the UK due to ash dieback disease. Felling of the trees is essential in areas where they become a danger to the public and hawkers, but we're also losing trees which have specific fungi attached to them. So we need to map the presence of all fungi within RCT. Also, it helps monitor global crisis. Species are shifting their geography. Species that you would normally find northernly are moving south, and vice versa. We are finding more and more within RCT species which are associated with the south of England. That is a combination of the fact that we have coastwise habitat, which is slightly warmer anyway and can mimic dune landscape. But it also is to do with global warming, because when you look at the overall shifting of fungi around the UK, then people throughout the UK are noticing species turning up that need warmer climate. And the concern is that when things keep moving south, where are our colder species going to go? So we have species which are already threatened with extinction, and unfortunately we are likely to lose species of fungal diversity within the UK. And finally, not least, it's vital for conservation and preservation. We can't protect if we don't know it's there. We haven't got enough people within RCT identifying fungi and certainly not recording fungi. So you don't have to be an expert. It's just as important if you recognise you're in a field that has wax caps, because by recording that you have a Hygrocybe, the wax cap family present, that may alert somebody to go along and do a survey. So every species, no matter how common it is, please do record it. And the next page, please, Rose. And where to record. Now, the Lurk app is an app that I find is pretty much fantastic for anyone and everyone. It's very easy to use on your phone or on a pad out in the field. It gives you your GPS and mapping straight away, so you don't have to mess around, find out where you are. It does it automatically for you. And it covers the whole of Wales, so no matter where you are in Wales, you're covered. Now, SuperCode, that is vital for our local authority decision making. This is the South East Wales Biodiversity Recording Network. This is where our local planners go to get biodiversity records. And if the records aren't in there, again, decisions are made to do with land management and planning decisions without the knowledge of fungi that may be there. And you may be talking about fungi that are exceptionally rare. They're associated with a tree, and once that tree's gone, that's it, that fungi's gone. We have very fragmented habitat within RCT, and you have clusters of fungi. So that particular species of fungi may not be found anywhere else in RCT. So, you know, it's vital that people get out, try to identify fungi, just have a go, you know, make those pointers, have a go, and there's lots of help out there to actually get you up and running. You can go along to Forest Local Morgan Fungus Group. You can use social media for the help and advice, and lots of good literature out there as well, which you can learn in your own time. So please just have a go and get recording. Thank you.
Brilliant. Thank you so much, Emma. That was absolutely brilliant. It blows my mind every time fungi, like you learn something new on every slide, basically. So thank you so much.
I hope I didn't go too fast for people. I was trying to race through about 20 minutes. It's like, I can't do it in 20 minutes. It's impossible.
No, sorry. It's a lot to cover in that time. But yeah, hopefully, because there's a recording, so people can go back to it as well. And yeah, I'll share my presentation again, as I've got, there we are. So that's got a sheet from the British Mycological Society on there, but their website is brilliant, isn't it, with loads of resources on it for ideas and things.
They've got excellent education site to the BMS as well. So yeah, lots of information available, and they're very supportive as well. So I'm obviously I'm a member. Glamorgan Fungus Group is an affiliated member as well. So they have some excellent learning tools, and they're getting better. Just in the last year, they have a new team on board as well, which are pushing education. So things are only going to get better. And just recently, fungi have now been officially recognised as being. So when we talk about flora and fauna, I've always said flora, fauna, fungi anyway, but now fungi is actually, or funga, they're calling it funga, is actually now recognised and being pushed now as well. So that it has to have a voice, because it underpins all ecology. So if you haven't got the right fungi, then you haven't got the ecology. No fungi, no orchids. You know, so we worry about preserving orchids, but yet we're not worried about preserving, like we'll chuck down glyphosate where orchids grow to clear an area for transplanting and so on, and we've just killed a microzyia or weakened them, which you know, those orchids, you know, they're vital. They can't even they can't live without those fungi. So very important. Yeah.
Oh, brilliant. Thank you so much again. Lots of nice comments in the chat as well, and I'm sure if anyone's got questions. Oh, Marie, if you got your hand up.
Yeah, so I just wanted to ask, it might sound like a stupid question, but you know, when you're doing, you're obviously looking at fungus under microscopes and stuff like that, what sort of things do you look out for then?
It depends. Sometimes it's simply splitting a species on the spore, on the type of spore, or the spore size and measurements of the spore. Other times it becomes much more technical, where we're looking at the basidia, which is the that's the fertile part that actually produces the spores and actually drops the spores off. The basidia can be absolutely wild in shape and size. Some of them, like in the jelly fungus, which are basidiomyces rather than ascomyces jellies, I describe them as being the octopus, because they actually have basidia with long tentacle-like arms coming off them, and they're actually amazing things to see. But we're also looking at pleurocystidia, so many features. And the unfortunate side of fungi ID is it is very technical. It does need an awful lot of high skill. You're not just looking for features, you're using chemicals as well as a microscope to enhance things, to get chemical reactions from a changing colour. Some spores will actually react with iodine, so they're amyloid spores. That with ascomycete in particular, we're looking at spores which turn, and the asci themselves, the actual the asci is the spore bearing body that actually shoots the spores off. That actually turns blue with that iodine. So yeah, lots of it is unfortunately very, very technical. But you can start off at a very basic level and just look at spores under a microscope, and you can look at. In fact, I keep hopefully when the brain's working a bit better, I'll actually I'm going to do a short video on showing the different microscopes, what their abilities are. So right the way down to a £5 microscope, all the way up to what I use, which is you know, a £1,500 microscope, showing what they can do, just so that it makes it affordable then for family to actually have a microscope that does the basics. That's enough, you know, that teaches them and sends their children off into the big wide world as well with the skill of being able to use a microscope, because they're not using microscopes in schools anymore, which is quite tragic. You know, children are entering comprehensive and then becoming students in university, and they've never put their hands on a microscope, which is it is quite sad. So yeah, hopefully I'll get around to doing that, but when the brain cells are working a bit better.
Brilliant.
Thank you
so much, Emma. That's great. There we are. We'll have to try and follow that now. So we'll just move on then to the next things, but thanks again. And yes, so back into October, so other things that you can do later in October. Well, you can do this in all the coming months now. So I just wanted to remind you about the dormouse nut hunting. This is a good time to do it. The hazels are just starting to fruit now, so you can get out there and have a good look and look for these nibbled nuts. This is a nice guide that shows you the different ways, and on our Facebook page as well, we do have a video that we made with the Wildlife Trust last year, which sort of explains a bit. That was in Brynner Woods. Brynner Woods is a really nice place to go actually for your first go at looking for dormouse nuts. There are dormouse there, and you won't see them yourselves. This one is one that had been caught in a box and went shooting off up a tree. So they are nocturnal, but you can definitely find the signs of them. So I'd recommend that. Is an absolutely lovely activity to do, and you know, every neighbourhood has got hazel tree in it, so it's worth going to have a look. And we don't know where they are. Again, they're like the fungi, they're quite under recorded, and we know we have a lot in the south of the county, good populations, but where else are they? It'd be good to find out more. So that's a really nice activity to do. And moving into November, then we've got our sort of more winter nuts and berries. So you can do nice hedgerow surveys, go out into the woodlands. And this is quite a nice worksheet to do with kids, a basic one, which tells you some to look for, so your hawthorn berries, holly berries, and so on. And it's a good chance to take out your your binoculars as well, and have a look for birds that might be feeding on the hedgerows at that time of year. And another one that's nice to push then is these sort of fruiting trees. Again, they're an important food source for birds over winter, so that's something to bear in mind for habitat improvements perhaps, or garden improvements. And November, October, this time of year is tree planting season. Obviously, we've talked about this in a previous Community Action for Nature workshop. So I've just put in again here the 10 golden rules for tree planting, which is that we look after what we've got already first. We work together. So for example, you know, you can come to us as the Nature Partnership and ask for advice. Select an appropriate areas, and that is a huge one. You know, we've just learned about all these important fungal sites, and that includes grasslands, that includes coal spoil, and that's a huge risk for coal spoil in RCT and all around Wales. Is remediation of these coal spoils quite often involves planting trees. Well, you might lose really important grassland sites by doing that. So that's a really important thing to bear in mind. Selecting the correct species is really important, and this is a nice activity that you could do if you are planning future tree planting. Is I think it's from the let me find my notes. From the September the 22nd to October the 22nd is seed gathering season. So if you are planning some future tree planting initiatives, you can go out to collect seeds from local stock this time during that period. So, you know, go and find your local oak tree, pick up some of the acorns from underneath. And this is a really nice, the treecouncil.org.uk. They've got a whole section on growing trees from seed, and that's all the different tree species as well. It's got a different worksheet for each tree species and how you can maximise getting them to grow from seed. So that's a really nice thing to do at this time of year. See if you can grow your very own oak trees. Volunteering is a huge thing at this time of year. There's not a lot to do survey wise, or there's not much wildlife out and about, but it does tend to be the time of year when most of the habitat management is done. For example, we're outside of bird nesting season. It might be a time when they're clearing scrub from grasslands. The Wildlife Trusts are a really good one. This hopefully, COVID permitting, there'll be work parties going out, but there's always opportunities to volunteer. So the Wildlife Trust are a good one. They have a number of reserves. There's South East Wales Rivers Trust. They'll be going out, groundworks, Keep Wales Tidy, all sorts of the organisations within the partnership. And it's worth looking for these local opportunities. And if you've got, you know, you can basically look for nature sites in your area and see who manages them and give them a shout and see if they need any help on their sites. That's always a good place to start. December, this is quite a nice, well, very nice thing to do. Digging a pond is one of the best things you can do for wildlife in an area. Even if I've put two options here, there's, you know, you can sink a mini wildlife pond, and that could just be a little container, as long as it's got a bit of water in it and some plants. It's a pond, and it's going to be really good for everything from amphibians to dragonflies. And they have benefits going all the way up the food chain then. So that's, December's quite, you know, if you pick a non-frozen ground day, December's quite a nice time to do it. It's an activity that people can get stuck in with when not a lot of other things are going on. So there's a few different worksheets here that tell you a bit about how you can do that. What is really important is thinking about where your plants come from. So rather, you know, I wouldn't go, if I'm digging a pond in Pontyclun, I wouldn't go all the way up the Rhondda Valleys to get some pond plants and spawn, because what you will find is, or what you might find if you're unlucky, is you transfer these sort of non-native invasive species. There's some horrible ones. Crassula is a terrible one which will kill the pond before it even starts, basically. And there are also some really horrible amphibian diseases that you can transfer unwittingly to your area, and it might be nowhere near, and you could accidentally introduce that. So that is something that you need to bear in mind. If you're going to move between ponds, make sure it's a very, very close pond, or you can source them from a reputable supplier. And there's one called Puddle Plants is a nice one. Another thing to note is you should avoid adding fish to your ponds. A lot of people sort of mention ponds and say they'll add a goldfish or two. Fish are one of the worst things for a wildlife pond because they'll just eat everything. So you'll lose all your dragonfly larvae, all your invertebrates, all your tadpoles as well. So avoid fish as much as you can. So that's a lovely one. And this is a nice Christmas activity as well. I really liked these photos with this sort of peanut chain. And you can make your Christmas decorations essentially out of bird seed. So this tells you a bit about how you can do that, but you can trial and error. I think this is American species, but a lovely Christmas tree outside decorated for the birds. So that's a really nice idea. Yeah, I know, I love the wreath. It's really pretty, isn't it? Made this when I was with the Wildlife Trust. It was a nice day. So yeah, I'd recommend, if there's a pine tree somewhere in the community or on a lawn somewhere, maybe you can see about decorating that for the birds, and that'd be a nice winter activity and serve the purpose of reminding people that it's important to feed the birds in the winter as well. So we're getting to the end of Nature's calendar. There will be one coming out now for next year soon, but I put it in again because these normally remain about the same dates. So just a little reminder. There aren't many coming up, but yeah, Wales Fungus Day, it might be around the 10th of October. There you are. But we've also got National Mammal Week at the end of October, again to be confirmed, but maybe that's a good one to send people out looking for their dormouse nuts. And Tree Week as well, so there's some good activities in there to do around Tree Week. Just a small reminder here about the funding pot that the Nature Partnership has. So it's a very small pot, but it does have up to, I think you can apply for up to £1,000 for community activities or community action for nature. So for example, I think the BTO, Wayne was here earlier, they've applied for some money for some nest box fund, to create some nest boxes and some equipment for bird ringing. So if that is something that you wanted to ask more about, just get in contact with me. That's absolutely fine. And I just wanted to let you know here about some of the activities and events that are happening with the partners of the Nature Partnership. So the Lost Peatlands project, which is an enormous peat bog restoration project, which is happening in the uplands of RCT and NPT. Loads of information about this online, but they're doing a huge peat bog restoration project with lots of community events, habitat management going on as well. They're going to be looking for volunteers over the winter, so they'll be a good ones to go to. Have they got there? Yes, there's the address there. It's lostpeatlands@npt.gov.uk. Over the next few days, actually, they've got open days going on at the habitat restoration areas and in the community wild spaces. So there are a few in RCT actually. So it's definitely worth going along to them if you can. It's a really interesting project. There's also going to be some online training that you can get involved with. So there's some reptile survey going on the 15th of September. So I think that'll be going out on their social media quite soon. Marie might be able to say more. Marie did our talk last, Community Action for Nature, about the roadside verd projects and the wildflower grassland management. And this site is live until when now, Marie?
The campaign is running until the end of September, and you can basically go onto the Let's Talk Wildflower web pages, which are illustrated there, and you can nominate areas of grass verges or patches of grass near you, which you feel could be left for wildflower management. And the plan is that once the campaign finishes, we will then go through the list, have a look at the ones that are feasible. Obviously, we can't guarantee that every single patch put forward will be actually maintained in that way, but we'll certainly look at them and assess them, and then hopefully they'll form part of the work schedule for next season, for next spring, summer, so to speak. This is not about, and I'm sorry forgive me for saying this, but this is not about us sowing wildflower seeds on grass verges. This is about us maintaining and encouraging the native flora that is in the seed bank through a different form of management. So rather than having your grass being cut every 6 weeks, these would be verges or areas of grass which would be left throughout the whole of the summer season, and we'll have one final cut or just one cut either in August or in September, where the clippings will be collected so that we can encourage more wildflowers again for the following season. But by all means, please check it out. We are really serious about doing this. We are committed to encourage more pollinators in our areas, and certainly to encourage more orchids, to use your words, Emma, earlier. So the more places get nominated, the more likely we are, we can increase that. And yeah, any feedback would be fab. Thank you.
Thanks, Marie. And as part of this work as well, we have got grab your rake events happening over the next few weeks. So the idea of these is that these are sites where perhaps the machinery, it's not suitable for the machinery for whatever reason, too steep or too small. And we're asking the local community to come out to these areas and help us rake the cuttings off. So we've got 2 in this Friday, 2 on Saturday in various areas. Marie, do you want to jump in again there?
Yeah, so Friday we are doing an area in Aberdare Park called Kaipu. We have a couple of banks up there which the guys are going to be strimming for us. So it's a case of please bring your rake with you. This is the whole idea of calling grab your rake, so that you bring your own rake with you if you can. So at this Aberdare Park in Kaipu, we're also doing on Friday Tallygarn grass verge. Again, similar sort of process. It's quite a small verge. It's by the roadside, but there's a lot of green wing orchids, I think, are growing on that verge. So we're trying to encourage that. And then on the Saturday, we're holding an event in Blenderondra Park. Again, it's an area of grass which has ancient ant hills, so we can't put a machine through it, otherwise we destroy the whole of the habitat. We did rake it last year, myself and the council of ecologists and some of the parks guys. but if we could get people up there, that would be fab. And we're also doing on the 4th, which is a Saturday, we're doing St. Defricks Hall in Treforest. There's an area of bank, again, it's quite a steep bank where you wouldn't get a machine on, where we're hoping to get more volunteers as well to help us doing some management. And then the final date then that we've scheduled is on the 10th of September, and we'll be in Handrevogen Recreational Grounds in Gilvarcourt. And this is a similar sort of setup, an area of bank that needs to be raked and with the clippings collected. So if you can spread the word, we are advertising it on social media. I'm hoping this week that the word will go out. It's on our website. It's also on the Let's Talk RCT website. So the more people, the more people know about it, the better. And as I said, it's about grabbing your own rake and your gloves and just having a bit of fun and help us doing some management that we wouldn't be able to do otherwise because we just haven't got the manpower to do it. So thank you for that as well.
Yeah, they're both, there's some questions about where they are and where and when. So they're all on the RCT LNP Facebook page as events. But do you want to put the email in the chat, Marie, for how they need to book it on and get more details, I guess?
What's happening is we obviously need to keep track of who's coming, who isn't. Well, not who isn't, but who's coming to the event. Just for COVID purposes, is nothing else. So if you could just drop me a line at parkscountryside@rctcbc.gov.uk, then I can send you the form then to fill in and give you more details in terms of location and time and that sort of thing.
Yeah, I can see you're not on Facebook. Sorry, Marie Claire. So I will send the details around as an email with the rest of this presentation as well. So I'll send that as an email too. So that'd be brilliant to see some of you there. Brill. So that's it for the evening. Look at that, right on time. So thank you very much everyone for coming. I'll stop the recording now so that you can turn your things on if you wanted to and have